Hey everyone, let's jump right into this explainer. Today, we're diving into something I like to call the hidden mechanics of cellular transport. Now, we all know that cells clump together to form these massive, complex tissues. I mean, they make up us, right? But for them to actually function cohesively as a unit, they have to constantly, and I mean literally constantly, negotiate with their environment. They have to take in the good stuff, the nutrients, and kick out the bad stuff, the waste. But here is a wild question for you. How exactly does a cell interact with the world when it doesn't even have a mouth? Think about it. For any cell to thrive, it absolutely must exchange materials with its surroundings. But without hands to grab food or a mouth to eat, how on earth does a tiny, single-celled organism pull off this life-or-death exchange on such a microscopic level? Well, the secret lies right at the edge. It's all about the cell membrane, or the plasma membrane, as we'll sometimes hear it called. This is an incredibly thin boundary that wraps right around the cell, and it doesn't just sit there protecting the squishy internal contents. It actually defines the cell's whole individuality. It acts as the absolute, definitive border between the inside of the cell and the great big outside world. Now, this border has a very specific rule. I like to think of it as the rule of the doorway. It is selectively permeable. Basically, it acts like a super strict bouncer at a club. It allows specific VIP substances to pass right on through while completely blocking others. Actually, we experience this all the time. It's exactly how oxygen and carbon dioxide effortlessly swap places across the membranes in our own lungs every single time we take a breath. But you know what? We don't even need $1 million microscope to see this invisible boundary doing its job. We can actually witness this whole process, this crazy selective gateway, in real time. And we're going to do it using a classic, foolproof demonstration with something you probably have in your kitchen right now, a simple, everyday potato. Yep, we're diving into the great potato mystery. Okay, so here's the setup. It's super straightforward. First, we take a potato and cut it into 2 roughly equal pieces. We make sure to weigh them first to get our baseline. That's absolutely crucial. Then, piece number 1 goes into a beaker of just plain, regular water. Let's call that beaker A. Piece number 2 goes into beaker B, which is a highly concentrated 20% salt or sugar solution. And then, we just wait. We leave them completely alone for about an hour. So, an hour passes and we pull them out. When we compare what these pieces looked like at the start versus how they look now, wow. I mean, the physical change is absolutely undeniable. Honestly, you don't even need to look at the scale to know something drastic happened. The shift in the overall size and squishiness of the potato structure is just glaringly obvious to the naked eye. So here's the breakdown. The exact same potato reacted in 2 completely opposite ways, dictated 100% by its environment. The piece that was just chilling in plain water, it actually swelled up. It got firmer and gained mass. But the piece sitting in that highly concentrated salt solution, it shrank, it got all floppy and completely lost mass. Which brings us to the real mystery here. What exactly is driving this dramatic shift? I mean, these are literally identical plant cells from the exact same potato. How could they gain mass in one beaker and lose it in the other? What invisible tug-of-war is happening here? The secret to the whole thing comes right back to that rule of the doorway, selective permeability. See, the potato's cell membrane is totally cool with water molecules. It lets them move freely in and out, no problem at all. But it completely blocks the larger, bulkier sugar or salt molecules. So the solutes are trapped on whichever side they started on. But the water is totally free to travel across the border. Now, water has a mind of its own. It naturally wants to find balance or equilibrium. So it will always move from a dilute area where there's lots of water and not much solute toward a concentrated area where there's less water and way more solute. It's basically trying to dilute the crowded party. So in our salt beaker, that super concentrated salty environment literally sucked the water right out of the potato cells, shrinking them down just to try and balance out the concentrations. And this very specific phenomenon, this movement of water pushing its way across a selectively permeable gatekeeper just to balance out a concentration difference, that, my friends, is what we call osmosis. But listen, this isn't just some neat parlor trick to do with a potato in your kitchen. This is a foundational, non-negotiable survival mechanism for life on Earth. Think about giant trees or the grass in your yard. Plants rely almost entirely on this exact process of osmosis. It's how their tiny, microscopic root cells manage to pull water directly out of damp soil. Literally, osmosis is how plants drink from the earth. No osmosis, no plants. Now, if we zoom out for a second, this whole crazy drive for balance is dictated by something called a concentration gradient. It's actually a universal rule. Whether you're dropping a tiny bit of blue food coloring into a glass of water, or someone sprays a bunch of perfume in the corner of a room, particles just naturally want to spread out. They move from an area where they are highly concentrated to an area where they aren't. This broader, overarching process, which by the way doesn't even need a membrane to happen, is called diffusion. So to pull the whole picture together for you, diffusion is just the general, everyday movement of particles spreading out down a concentration gradient. Osmosis, on the other hand, is like a highly specialized VIP version of diffusion. It's specifically dealing with water, and specifically about that water navigating its way through a cell's selectively permeable boundary. All right, to wrap up today's explainer, I really want to leave you with a fun, real-world puzzle to chew on. Let's say you take some dry mung bean seeds and soak them in plain water overnight, like for a full 12 hours. Thanks to the magic of osmosis, you wake up and they are completely swollen, firm, and packed with water. Now, what if you take those beautifully swollen seeds, pull them out of the plain water, and plunge them straight into a ridiculously concentrated salt solution? Based on the ironclad rules of osmosis we just went over, what exactly do you predict is going to happen to them? Think about where the water is, think about those selective boundaries, and think about that unstoppable drive for equilibrium.